overall head count is going to be added. Second part I wanted to highlight here is the impact of increasing temperature on the crop yield. This I have adapted from the Zhao 2017. Very wonderful work he has done along with other colleagues. He has clearly, you can say, estimated the, you can say, reduction in the crop yield and the different parameters. So I'm not going to much detail on that. I'm just going to say summary of that, that each 1% increase in the temperature will impact yield of rice by roughly minus 3%, wheat roughly 6%, corn 7.4, and soybean 3.1. And the trend is almost similar to major other food crops. So two aspects here we have to think. On one aspect, we are adding more, you can say, people on earth. On another side, because of increasing temperature, we are going to lose, you can say, percentage of our production on that way. So it is you can say very challenging situation. On one aspect, population is increasing. On another aspect, you can say our yield is going to increasing. Although we are adding, you can say in terms of food production, lots of things, but yield increase is really very challenging in that way. So this is the, you can say, current scenario at the moment. And as a breeder, as breeder, everyone here or any crop scientist, their first task ideally should be like how we can maintain that crop production versus the growth of population, that thing. And in this scenario, breeding comes in the picture that shows the silver lining. And it is not that today and tomorrow it will show, even past also it has shown that thing. And that is, I'm going to say in next slide. Before that, I just wanted to again say a few of the facts about, and I'm going to share the what plant breeding has done. So as I mentioned, 2.2 billion more people are going to be added by 2050. 50% of the more food and feed is required to meet that demand. 3 billion people will live on agriculture areas with high to very high water shortage, and that is very challenging. 70% of the all available fresh water we are going to use in agriculture currently. And on other side, there is, you can say, pressure on the ecosystem. Either it is crop loss, as I mentioned, temperature alone is going to have roughly 8 to 10%. Adding to that, other biotic and abiotic diseases that is going to, you can further decline it. 90% of the all soil are expected to be lose their quality little bit or different extent by 2050, and 20% loss in arable land per capita because of industrialization and other, you can see, land use. This thing will happen in future. So seeing that this complete scenario, then this plant breeding come in the picture, and I just wanted to say this slide, how the plant breeding has helped significantly. And when I'm telling plant breeding, it is not alone only breeding here, rather it is, you can say, crop management also that help in that way. On the one front, you are seeing from 1961 to currently, population is increasing very, you can say, its sterility is increasing, but at the same time, plant breeding has also, you can say, provided productivity of all the food crops. It increased roughly from 200% over the last 60 years. And that also resulted in the cereal production by roughly 250%. So that is where our breeding efforts and other associated, you can say, efforts from other functions really led to, you can say, coming on that front. But here, one thing that is very challenging, which we find, the food security is not only availability of food to person there, something like that. There are many other things. It's affordability, availability to each and every person on the earth. And another thing which generally we forget, if you see the FAO data or UN data, the gender disequality in the food security is always there, where our women colleagues having little bit more food insecurity as compared to their male counterpart. And it is across, you can say, continent and global level also. So on that front also, we have to, you can say, think it of going forward. So I wanted to here emphasize on that, even though challenges are there, but plant breeder in our other functions keeps on doing new innovations and then keep the productivity at the pace that is helping us you can say, feed this world population. And we have to keep that pace in the coming, you can say, future also. And then when as Dr. Ilyas mentioned that there are many innovations happens over couple of centuries there. So if I divide this evolutionary journey of plant breeding, I can find there are clear 5 phases. One is the domestication and imperial selection, starting from roughly 10,000 years back, when majority of selection happened by you can say our ancestor. Second phase comes roughly 1900, when we rediscover the principles of genetics by our father of genetics, Mendel, and we started typical crossing and other population breeding. Third thing came Green Revolution and then mutation approaches, when our you can say short stature varieties of wheat and rice came in the picture, and then they revolutionized our production. Fourth phase came roughly in 70s, when we you can say fully understood the you can say DNA structure, as well as the all the different technology, this marker assisted or DNA technology. We can tell it is a gene revolution at that time. And last stage, that is couple of decades back, or you can say first quarter of this century, where we came more about this understanding of our genomic structure and a new technology in terms of the, I can say, digital farm. Then next thing came in the picture. So these 5, you can say, major phases of breeding, that is, I'm going to share few of the glimpses of those innovations that happened, especially in the last one. So if you see here again, the same 5 phases. So in first phase, it is mostly on the, let me see the pointer here, so on the domestication and empirical selection, mostly it is domestication, and it is done purely on an observable phenotype. However, in classical and Mendelian population, in population breeding, it is we started our pedigree selection, recurrent selection, double cross maize hybrid, and even we are started exploiting heterosis and cytoplasmic male sterility. So that is, you can say, one of the very crucial milestone or innovation in that phase. Third phase, that is Green Revolution and mutational approaches, and 1940 to 60, this semi-dwarf and fertilizer responsive wheat and rice variety that is really revolutionized at that time. Dr. Borlaug and led that thing, and then we tell that era at the Green Revolution, and in India, Dr. M.S. Swaminathan and a team and many other stalwarts of agriculture, you can say, came in the picture and really did wonderful job on that. And in same time, mutagenesis also took a place, and then where we developed few of the varieties of, you can say, through mutagenesis, and new alleles of trait we have generated. Coming to 70s, when we completely understood the DNA structure, as well as how it is behave and how we can little bit, you can say, modify it, then recombinant DNA technology and agrobacterium transformation, and later GM crops enabled direct gene transfer came in the picture. And then there are many GM crops that is currently having across the globe. And we can tell that it's gene revolution that happens there. At the same time, tissue culture, double applied and molecular marker also came. RFL, PSSR, and SNP, I'm not going to detail on that thing, but these are the things that keeps on coming, and they are still relevant, few of them. Last but not least phase, that is genomic speed and digital breeding. It is last this quarter of this century, whole genome sequencing, GWAS, genome selection, and genome editing that is came in the picture, and that is now, you can say, center, you can say, cornerstone of current, you can say, crop improvement, this thing that is happening at different level. And then further, these are coupled with the speed breeding, high throughput phenotyping, and then data science and AI, and those things are happening at the moment. So I'm not going to cover all of them. Majority of things I'm going to cover only the last phase of this thing, and then few of them, what is really having very high stake as well as giving result, I'm going to discuss on that front. So I have identified, rather selected these 4, you can say, genomic selection, gene editing, speed breeding, and precision phenotyping, including digital twins, for today's presentation. So coming to genomic selection, Ed, I believe if you are plant breeding and recently you can say taken the plant breeding course, I believe this is one of the component. But when we used to be a student, then this was not that much popular, or we are not aware about this thing because that has not came in the that you can say detailed part. So genomic selection is a very powerful breeding strategy that uses the genome-wide genetic marker like SNP and a statistical model to predict the breeding value of individual, enabling faster and more accurate selection for desirable trait in livestock and plants. It is equally for both. And one thing is a very good thing here, work that it works by a training model with known genotype and phenotype to predict the performance of untested individual. That is one of the thing. And if we come in the how it works, there are typically four stages or phases of this in the genomic selection. I'm not going to detail of each and everything, just giving the glimpse of that. The first thing is data collection. Mostly we do the high density marker data, maybe hundreds or thousands of SNPs and phenotypic data that is we took in the field for different trait like yield and diseases that is collected on large training population. And then based on that data, machine learning and statistical model, mostly blobs, we use to estimate the effect of all those markers at the same time and build a prediction equation. Once prediction equation is built, then this model, you can say through this model, we predict the genomic estimated breeding value, that is GEBV. And then based on this, all the, you can say, best breeding value, we generally select those individuals, and these selected individual based on the highest GVB are selected for the next breeding cycle. That is accelerating the genetic progress. Key advantage of this genomic selection is the increased accuracy because we are using you can thousands of marker, and even it is equally good for a complex trait where we can have difficulty in conventional breeding methods. Reduce time because it reduced time because we are doing this selection very younger ages and even before traits are fully expressed. Cost effective, although initial cost is a bit high, but once it is fully developed and even with the third party also, we minimize costly and time consuming and the field and lab phenotyping. And its application is, you can say, almost all type of trait, either it is highly heritable or even low heritable traits, both it can work well. And if you see the application both in plant breeding and animal breeding, so it is equally, you can say become applicable, and it accelerates the development of crop with high yield, better quality, and improved stress tolerance. So this is one part. Another thing that is coming in picture, that is one technology or one tools we are using in genomic selection is gene editing. So in genome or gene editing, it is nothing but it is a group of technologies that we use to you can say change the DNA of an organism. And if you see the left side, this is all type of you can say methods are there where we change the DNA structure like distant hybridization, conventional breeding, tDNA mediated integration, mutagenesis, and the other like recombinant system. And here it is it is the, you can say, genome editing part, this genomic selection part we can. So in genome editing basically use the JFN, TALEN, and most commonly CRISPR-Cas you can system where we can do it very precisely, change the DNA. There are three types of gene editing that happens: SDN1, SDN2, and SDN3. SDN1 and 2 is coming in the very you can say very normal way of doing where regulation is not required. But in SDN3, where it is we are taking the gene from some other organism, there it is treated as a GM crop. But SDN1 and SDN2, where it is we are just changing the structure within the DNA of organism, that is not required any type of GM, you can say, enabling on that. And it is very easy to do. Not easy to, you can say, get regularized and getting the regulators approval. And if you see the genome editing across crop for last, you can say roughly decades there, maximum this editing done in the rice, that is 50 and 37, followed by maize, wheat, barley, and other crop. So rice, since its sequencing is complete, we know the complete sequence of genome sequence of this thing. And then because it is very, very good, you can say, crop to work with the biotechnological tools. So I think that is one of the reason this crop has been used multiple times for this genome editing for different traits. But other crops also has been done, mostly for disease resistance, weed resistance, and then even nutritional value also, and even some of the other crops in the horticulture also. This genome editing has done well. I'm not going to tell you can say detail on that thing because everything is available on net. Even I have collected few other information on the open access, so I have given reference also. You can refer it and then get more you can say information on that. Second is speed breeding. Third, you can say our innovation that is currently happening. And then why speed breeding is so important? This is the, you can say, region you find here. If you see the genetic gain formula, that is what genetic gain we are having, it is heritability multiplied by the variance into the selection intensity divided by genetic generation interval. So this is the why that plays a very important role. If you are having all the top level things constant, even if you reduce the generational interval, still you can get the more genetic gain in these sequential, you can say, generation. So why is the here that is letting the you can say breeder and other technology savvy colleagues to think it off? And if I'm correct, it was done in Australia first time, this speed breeding, and they are doing multiple crop, and now I believe it is across globe. This speed breeding is being practiced to do the, you can say, multiple cycle of the crop in the one particular year. So once you reduce the cycle of time, then definitely genetic gain will be more, other than the other factors. So if you see the here, sorry, it is overlapping. No, so speed breeding, if you see, it is the now become a central tool in modern rapid generation breeding strategy. And if you see this part, it is wonderful thing if you really want to make the progress in the genetic advancement. So major of you can say speed breeding is the suite of controlled environment technique. Basically, we do in greenhouses or you can say glass houses, basically, to shorten the time between the generation. As breed and we can grow multiple generation in 1 year. And in this speed breeding, generally we control the photo period, light intensity and quality, temperature, and sometimes carbon dioxide also. Typical protocol for you can say in speed breeding is we extend the photo period for 20 to 22 hours for long day crop, and then roughly 10 hours for short day crops there, and that allows us to have 3 to 9 generations per year, depending on type of crop, as well as what type of you can say objective you are going to have. We generally keep temperature in roughly 22 in daytime and 17 degree night, but it varies from the crop to crop. And another thing we do in the speed breeding is rapid drying after maturity, so that we can get the seed very quickly and then grow for the next season. Speed breeding also combine the it can combine the single seed descent method, genotyping part, genomic selection, and gene editing to accelerate the line fixation, as well as trait integration and testing pipelines. So it is just you can say it is speeding up our cycle per year. And I'm not going to detail on the other part here. It has been overlapped by another photo. My apology for that thing. So it advantage is, you can say, better than the our subtle breeding or glass house, a greenhouse production or something like that, where it is multiplying multiple generation in 1 year. Limitation part here, it is basically the little bit, you can say, early time. It is a more cost included. Once it is done, then it will become quite, you can say, very, very easy and very cost effective. And if you see the different, you can say, example of this speed breeding, I have given the multiple lists taken from the Syamala and Cypripedium recently. They have done the work. They have collected this information. So all type of crop, either it is long day crop or short day crop, both are equally, you can say, impacted by this. And we can get the 4 to 9 generation depending on the type of crop in this speed breeding. Last but not least is the precision phenotyping, including digital twins. So digital, when we talk the precision phenotyping, it doesn't start only when we take the data in the field. Whether it start planting and planting time also, in season, and even harvesting time also. And then to ensure those precision phenotyping aspects, there are many digital interventions are available. And currently, many of the companies, means private sector and public sector, are using it, you can say, profusely, to have, you can say, better data quality. So in pre-planting and planting time, generally digital field mapping, generally we do in the Bayer, and I believe it is being done multiple, you can say, other organizations also. So digital field mapping is done only to have ensure that our trial is planted at particular place, so that when we are having the image either by drone or by satellite, it can accurately, you can say, pinpoint that thing. And our planting generally we do with the precision planter that is GPS enabled, and then here we can say absolute, there is no deviation even for 5 centimeter also. It cannot have deviation in the road to road distance or plant to plant distance. So that is precision planter, where another intervention is helping us in the precision phenotyping. In season, if you see, there are two aspects. One is vegetation and soil sensors that is giving the real-time data on the our crop growth and then whole season, and soil sensor are giving all the you can say parameters that is for soil, either in terms of you can say NPK or the moisture in that sense, and it is helping a lot in the optimizing the resources. Second part in season is that is we are taking the data through UAE and satellite also, and its application is altogether, you can say, very apt to see the real you can say quality data, because with satellite also we can go for the like maturity characterization of the crop, where your field of the trial can be captured, image can captured by satellite at a regular interval, and you can really assess your you can say maturity of the crop based on that image, even if you are not you can say going the field. But ideally, as breeder, we have to go. But sometimes you have thousands of the trial. So this type of intervention will help save lots of time and efforts there. And last but not least, harvesting, where we are using the precision harvester also, where it is just you have to run the machine in the field. All the data, required data, will be collected and then transferred to the system automatically. There is no human intervention in this. That is helping us in the, I can say, errorless data collection on that front. So all these things are, you can say, in the crux, we can say, these digital intervention for precision phenotyping is helping to ensure the scale, speed, and accuracy for quality data in time. And that is very much required to intervene all the new innovation, because all these new innovations based on the huge data that is on that they keeps on churning and modeling, and then based on that we take decision. So quality data is very important for that. One extension about this for digital phenotyping, that you can say it is a little bit more on the digital twin there. I know that majority of you know digital twin, and if you are not knowing, I just want to give a very brief about this thing. Upon how I'm carrying the time.
You're okay, please continue.
Okay, thank you. So on the digital twin, it is digital twin is the simply virtual representation of an object system that span its life cycle. It can be any object, it can be any process, it can be any asset, and it can be any system. And even it can be any part of the, you can say, your breeding program also. There are 4 major components of digital twin. One is the object and system, as I mentioning. So for agriculture, I can say our trialing location or sites is the one of the object of that thing. Sensor and imagery, as I mentioned earlier, sensor took all the data or parameters at particular location, and imagery take image of the that object or system at regular interval, and it is continuous process. Third component of digital twin is the Internet of Things. It is like all the connected devices via internet, so that whatever data is being collected regularly, they keeps on moving into the system without any human intervention. So it keeps on continuous flow on that thing. And fourth and most important thing is simulation and modeling. Once we have enough data of a particular, you can say, object, then we can start simulating it and developing a model that will give the, idea about how the digital twin of a particular asset or our field is working. For example, I'm just giving something much, you can say, simpler way. If it is too technical for those colleagues who are new. For example, if you are sitting in the Banaras or Varanasi and you map all the weather components there at particular location, and the similar way, if you are having the all the things of environmental parameter at the maybe in Europe, at certain places, maybe in France, suppose you can say, Paris, you can say. If both places, all the environmental components are matching over the long period of time, then we can say Varanasi and Paris is the digital twin in terms of the environment, or in terms of the field, or in terms of the trial location. So if I'm doing any trial at Varanasi, I can confidently tell, even though I'm not testing that thing at Paris, still I can say that genotype tested at Banaras can perform in the Paris also. So that is the digital twin. And if you see it the little bit larger level, if you are having 500 trial location and you have complete mapping of these trial location through this component or digital twin part, then we can identify out of those 500 locations, we can conduct the trial only at 100 location, because the remaining 400 are matching with one or other in that sense. So it is something like, you can say, optimizing the resources and making us more, you can say, efficient. And it gives also a concept of prescriptive testing, where we can prescribe, if I'm planning to evaluate certain product at certain geography, we can tell that in spite of going 100 trials, guys, do the only 10 trials in these locations. It is going to represent the whole area in that sense. So that is the, you can say, in very, very nutshell about digital twin. It is mostly used earlier in the aerospace industry, medical science, but in agriculture, it is little bit, you can say, new, but many companies are working on that thing, including Bayer, where we have already implemented it. So little bit more detail on that front, like digital twin in agriculture create real-time virtual replica of farm, crop, livestock, or equipment. And then if you see this, what it does basically in crop management application, it can monitor the crop growth, predict yield, and optimize inputs. Another thing is can reduce the wastage of 20 to 30% of water and chemicals, because it is you can say, measuring the soil moisture at regular interval, and at particular threshold only, then you have to apply the water. And even we have done in our, you can say, internally, and we find there's roughly 30 to 40% water can be saved if you are really using it properly. But scalability is one of the challenge, because it can be done on our research things, but on the larger scale, that is a little bit challenging in the current scenario. But going forward, I believe once service provider will become in the, you can say, in majority, then I think it is going to be help not only the research, even the you can say commercial agriculture also. On sustainability aspect, it can, you can say support by scenario testing like drought or flood, or even salinity also, tracking carbon footprint, adding a compliance and resource efficiency. And in supply chain, they can forecast post-harvest losses and optimize logistics from farm to market. So these are the few of the application that we can see. And still in crop breeding, like what we discussed in the genome editing or genomic selection, this can simulate the GI-environment interaction and accelerating evolution of edited or speed-bred lines under virtual stress scenario before actual field trial. So that can save lots of time and efforts, but it doesn't exclude your field testing. But here, speed is going to help us a lot in that way. So that is the way a digital twin is, you can say, we are using in the, you can say, agriculture. And then yield prediction is another one aspect where we can keep in mind. Coming to sustainability and regenerative agriculture, I'm mostly going to concentrate on what we are doing within Bayer, because That is where we are you can say, currently, you can say, progressing on that front. Two aspects I'm going to have here. One is the sustainability, and there is a regenerative agriculture. Although these two terms are very close to each other, but in principle, there is a bit difference that is coming in the picture. So Bayer is very much, you can say, forefront on this front. Their focus, sustainability focus, we are not losing, but we are giving more focus on regenerative focus, agriculture also. So if you see the punchline for sustainability, produce more with less, but in regenerative agriculture, it is producing more and restoring more. So we are restoring also, so that it can be, you can say, more, you can say, sustainable for longer term. And then one thing is differential here in the regenerative agriculture is we are delivering nature positive outcomes by improving soil health, restoring the biodiversity, and protecting the habitat. And then it is on larger front, but at a crop level or chemistry level, where Bayer is doing, they have clear cut their target for 2030, seeing all the 6 major global challenges for water quality, soil health, climate change, sustainable energy resources, growing population, and increasing protein demand. So these 6 major global challenges. So what Bayer has, you can say, committed by 2030, we are going to reduce the crop protection impact by 30%. Greenhouses get in the field production at roughly 30%. 25% reduction in water use per kg of crop produced in the rice cropping system, and then empowering roughly 100 million smallholder farmer by 2030. And we have already achieved more than 55 million through different, either through innovation or through digital outreach. We have already, you can say, connected with 52 or 55 million smallholder farm, mostly in Asia. Our target is to engage them, roughly 100 million farmer engaging them. So there are many solutions Bayer has at the moment, but I'm going to talk only at 2 aspects. One is the Preceon. If you have not heard, I will share a little bit more detail. And then there is Arise, that is the hybrid rice business brand of rice. So on Preceon, it is nothing but sod corn. And in Arise, I'm going to discuss more on the direct seeded rice. So in direct seeded rice, you can say Bayer has committed to, you can say, transform this rice system to completely to regenerative format, where earlier, if you know, it is still very much prominent, this transplanted rice that is quite resource intensive, where we need to do lots of puddling and labeling, and then lots of water we use there. Then go for nursery bed, again, we require lots of efforts here. Then manual transplanting, although there is a, you can say, commercial transplanter is also there, but that is still penetration is not that much. Machine or manual reaping, that is one thing. So these are the, you can say, few, you can say, labor resources intensive exercise we have in the transplanting rice. And then Bayer wants to convert this resource incentive transplanted rice into technology-driven direct seeded rice through laser land labeling, direct seeding with machinery, because there are many machines is now available, and there is precision, you can say, machinery also available. And you can say, ensuring the emergence of rice crop through different new technologies and mechanical harvesting. So these are the, you can say, Bayer's commitment, and we are focusing more on the direct seeded rice system at the moment. But our, you can say, earlier efforts on the TPR, we are maintaining it, because that is still, you can say, it required in the larger geography of country or region at the global level. So how this DSR is, you can say, helping us in the regenerative agriculture. So as far the yield increase and improve productivity, so I can very clearly tell at the moment, DSR is either little bit giving lower yield than the transplanted. It is well-known things, but we are working to infuse the new technology on that thing, and then at least make them at par with the transplanted rice. It is going to reduce roughly 50% of the manual labor. That is going to reduce 15 to 25% of lower cost, improve soil health because of not much disturbing on that thing. And generally, it started the crop duration by 7 to 10 days. That is, again, going to save on the carbon footprint. And when it is not transplanted, then definitely puddling will not happen. So that is going to reduce the greenhouse emission up to 45%. And here, one thing I would like to emphasize, because rice, mostly it is methane emission, and that is roughly 25% more toxic than the carbon dioxide. So in that way, this direct seeded rice is going to help us more on that front. And conservation of water, as many you can say reports, it is going to reduce roughly 40% of the water as compared to transplanted rice. But one thing I would like to clear very much here, transplanted rice is still, it is going to be, you can say, one of the things at present. Slowly, we are working on that. How fast we can adopt this technology, and then we can leverage the what benefit we are going to get. It cannot be done within 1 year or 2 years something. So and then also we have to ensure the food production also in that way. So these things will keep parallelly, and then as and when and more technology will come, we'll keep on enabling this new way of regenerative agriculture through DSR in the rice in that way. Another very good example of you can say integration of complete new genome editing, or you can say genome breeding, genomic selection, as well as the sustainability part. Bayer has developed the smart corn system that is known as Preceon. Preceon is nothing but it is salt corn. That is we have you can say launched 2024 in the North America and 25 in EMEA. And the later we are going to launch 2027 in Asia and 2028 or 29 onwards. So major, you can say, benefit of this hybrid is that it is giving the protection again high winds and challenging weather condition. And access here, because access is one thing that is not applicable in Asia at the moment, but going forward it is also going to happen. We are providing the you can say in season you can say access to different forecast, as well as the technological help to our bigger farmer in the Latin America and North America through Farm Rise, one of our you can say data driven or digital capability things there. Yield potential, it is definitely going to optimize input also, and planting density we can have more, and in that way yield potential we can maintain. And these two aspects, silage and sustainability, it is very good for silage actually, and silage is very common in the you can say Western world, including Europe, and also it is now taking up the you can say importance in the India, where many you can say companies are coming for silage production and distribution to dairy farmers and even the different FPOs also. And it is going to reduce the greenhouse gases also, and it is going to help us in the carbon sequestration also. And we are working with different external stakeholder for this hybrid to further add value on that thing. And currently this is on you can say we having with BASF also some you can say collaboration to add value on this Preceon. So these are the few things I just wanted to share on the sustainability part there and regenerative agriculture. This is my last slide. Way forward, what we want to do and how it is going to happen in the future. So I'm not going to tell anything new. It is just whatever information is available on the globe or on our community. So first and foremost thing I want to share here, our old breeding system or conventional breeding system, these technologies are not going to replace them. Rather, they are going to add them, supplement them, and support them. And then it is just like you can say putting the integrated you can say approach for these technology with our you can say customer requirement. That is one of the first thing we have to you can say take. Second thing is the early breeding wisdom, what we have learned so far in conventional, and new breeding innovation, what I talk about, and even beyond there many more you can see innovations are there. And the big thing already taken stage, but also coming well, and the data science and artificial intelligence and machine learning. So you people are now seeing the many things on that. So in nutshell, I can say convergence of these four elements, and then the as Aparna mentioned earlier, policy, you can say policy part from government level, and then accessibility to you can say food, and then other you can say things to our smallholder farmer is going to be one way where we can you can say, ensure the food security and sustainability at the country level, regional level, and even at global level. So this is brief about this thing, and I am open to for any question and other things. And before that, again, I thank you again for each one of you for sparing time, and again, ATP BVR, as well as Dr. Ilyas Ahmed for what there, you can say, moderating this thing. So this is from my side. I can be reached out at this email and also on the LinkedIn, and also not very active on Facebook, but LinkedIn I can be connected at any point of time. So again, on behalf of myself and on behalf of my organization buyer, I really thank you for giving me opportunity and hearing me. Over to you, Aparna, and then Dr. Ilyas.
Thank you. Thank you very much, Dr. Shukla. This is Suren Tiku, also from ATPBR. I think Aparna may have had some work. She may have probably left. I request Dr. Ilyas to kind of moderate any questions and answers that we have. Dr. Ilyas, you are there? Dr. Ilyas?
Yeah, I'm here.
Yeah. So I
Hello.
think maybe you can help the Q&A session and then we can conclude. Dr.
Yeah.
Aparna had some urgent thing to attend to, so she may have left.
Okay.
Thank you, Dr. Shukla, for an excellent talk. Yeah, I do have a few questions, but let us hear others first.
Now, how do we? There are so many people who want to ask questions. Do we?
Yeah, I mean, we can spend 10 minutes for.
Yeah, I think we have to have some limit.
Yeah, 10 minutes from if everybody agrees. There are some questions in the chat box. Could you take them or shall I?
Yeah, I think you are much more experienced in this line. Questions you can select.
Okay, I'll just select a few questions.
Yeah.
There is Grashneshwar Garkar.
Yeah.
In speed breeding used for line development is speed breeding used for the line development by breaking hybrids? Does selection under a speed breeding controlled environment have any effect on line selection when evaluated under field conditions? That is the first question, Dr. Shukla.
So as I mentioned in the speed breeding, it is we don't do any selection in the when we do the speed breeding in the greenhouse. It is just for sake of increasing the cycle per year. And different and it depend on the type of your objective as well as crop also, how many cycles you are going to do and what level of F1, F2 or F3 you are going to do this thing. And thereafter, definitely it is going to field. Actual selection going to happen there, but early generation we do in the greenhouse, glass house, in that's the controlled environment.
Yeah. So I think for me, can I add a couple of points there?
Sure.
To me, speed breeding does two things. One, it can do quickly integration of new traits into a current inbred. So from a donor, you can transfer a trait, but much more quickly, you can in 1 year actually get a new inbred using the background and foreground selection, because genomics are available in most of these crops. So that is one. The second part is, I think, as you said, multiple generations, we can do an SSD. Maybe you can select your F2 plant in the right environment, and then in SSD, you are in F6 within 1 year. So you are basically controlling the time taken to the genetic gain. That's what you mentioned in one of your. So I would just add those to what you have said. Thank you.
Yeah, I would add one more point,
yes,
which
please.
is most important. We don't lose any variability from F2 till F5. This is most important from genetic point of view.
Yeah.
And we have the genetic means by biotech and all to look for the trait we want. Entire genetic diversity is available from the original class.
Yeah.
So this is the main advantage of SSD and coupled with the speed breeding.
And also, I think you can also create your RILs within a year, and you are ready for your marker analysis, etc.
Exactly.
Right? Thank you. And then there is one question from Bapu Devkate. In genomic selection, how many environments should we use for developing a training population? And for how long can a training population remain effective for prediction?
Oh, that's a great question.
Yeah, that's a good
So,
question.
how many environment it is, you can say depends again, I think, type of crop as well as what is your ultimate objective. Okay, based on that, you can define the environment. Ideally, environment here, because majority of things we are doing the marker, you can say thousands of marker in that way. So on the typical genomic selection, there is basically not involve any physical things, you can say in that way, but you need to training population continuously and keeps it on the training year after year, because you are going to add some new xeroplas, new genotype selected there. So it is continuous process. And depending on the, because if you are experienced on that front, you know how the value these are adding to the new lines are adding. So it is continuous process on the training population. And then more the time you can say do the training population, add the new plans there based on their GEVB, then definitely it is going to be add value there. Whosoever asked this question, but
Thank you.
there is no, yeah.
Bapu, you have any clarifications? But I just wanted to, because I also have a question which I have faced in my life. Because let me talk about bacterial wilt in tomato. So we have various strains that have differential response in different locations. So if you are doing a marker development, so do we need to develop F2s and screen them in these different hotspots, and then look at the genomics in these hotspots separately, and then probably predict an overall QTLs that are, is that the right approach or not?
See, well, see again, as I told, it depends on the type of crop,
Yeah.
what you really want to achieve.
Yeah.
And then single strategy or single technology will not fit every, you can say everywhere.
Exactly.
So
Exactly.
it again depends on the type of crop also.
Yeah.
For rice, we are doing in the, you can say single environment, and then it is helping us a lot there in that way. But depending on our, you can say business requirement, we keeps on changing that as in time we require. Suren Garu.
Okay. Thank you. And I think, can someone explain what exactly is digital twins? You have explained, but maybe a little more simpler way. Can
Okay.
you? Yeah.
Okay, I can take it more on that. So before going into typical digital twin in agriculture, because it is new, just take your aircraft. Like two companies are there, Boeing and an Airbus. So most of plane in which you are flying, all of them having digital twin. Means one digital, you can say a replica is always there in the system. When you are flying in that plane, and you might have seen many sensors that is on the plane there. So
Yes.
each and every moment data is being captured in your digital system, and they are seeing it. And regular data is collected for that thing, and that will help them to further refine it. Where just some challenges is coming or something like that. So it is complete replica of one physical asset that is digital, where you keeps on getting the data through imagery and sensors, and then adding or simulating it to get the better outcome of that object. And in agriculture, as I told, for example, if you took your field testing location, it can be digital twin of another location based on the similarity in the environmental factor, either it is temperature, rainfall, humidity, soil type, and many other micro elements there. And for that, two things, as I mentioned, two things are very important. You have to capture data either through some sensors, or through images. So those things require to develop a digital twin of a field testing. So in that way, as I told, if you have hundreds of the you can testing locations, and if you have complete mapping of all those 100 locations, you may find out of that out of 100 locations or testing sites, there are 20 sites who are exactly resembling with each other in terms of the environmental scenario. So you have not to plan the trial on those 20, you can say, locations. Even one or two will serve the purpose in that sense. So remaining 18, you can just exclude. And even if you are not testing trial there, still you can predict the value based on your these, you can say, one or two locations where you are doing. So
Yeah.
I hope I have
I mean,
clear more.
especially for those where the environmental profiling is same. Then you don't have to go to every, but if the environmental profiling is different, then you may have to identify those spots so that you can then correlate the data. So thank you. I think that's very clear. I think before I hand over to Dr. Ilyas to conclude, I wanted to just add, I am also a plant breeder, and you said, that plant breeders have done, I mean, you have shown in your, what we have done in the last 2 centuries, but I feel we should also acknowledge the contributions of the pathologists, the entomologists, the physiologists, and the genomic scientists, without which we breeders, I feel, I have always felt that we breeders are like chefs. Only the good chefs get Michelin stars because they know which masala to put. So we translate the research of others into a tangible, commercial asset. So that's what good breeders do. And of course, they use their own innovations too, but it's a teamwork. Thank you. And I think Dr. Ilyas, please, please conclude.
Yeah, it's a pleasure to conclude the topic. First of all, I would like to congratulate Dr. Shukla for his excellent presentation.
Yes.
The most important thing was he kept within his time, which I liked. And he made it short only to 5 major points. I liked it. And however, I have few comments on each of these. I will not take much time, but I think it will be interesting. I will just give my comments. As I said, I liked his time management, his slides, but I would have preferred if he had less written matter on the slides than so much. Then it becomes easy to read. If you have 4 or 5 lines and convey the message, I think that is more effective for the audience because no one has time to stare and try to read. And by the time you read first 2 lines, the last 5 will go.
Yes.
Now, what effectively happens is have only 4 to 6 lines, bold. You explain. That is more effective way of transferring the information. That's what I feel by my experience. So I always keep 4 to 6 lines, very bold. So that is one suggestion. And he rightly began with population growth. Wonderful. And from US Census data, actually per 1 hour we have been now hearing, within this time 10,500 people have been added worldwide.
Yes.
2 lakh people per day.
Yeah.
25 data.
Yes.
So this is the speed at which we are increasing. Yeah, and the temperature, effect of temperature on yield is also very clear. This year, the climate has been very uneven in India, in Middle East, US, even Ukraine and Russia. There are a lot of crop, yields have gone down. If you see latest data, global picture, you will have still better data where the red lines, red figures are coming. The moment temperature is going up, the production is coming down. Contributions of plant breeders, of course, is a very big topic. And we plant breeders always feel we have contributed more, but very sensible comments from Suren Tikku that everyone, team is involved. Yes, team is involved. Yes, I think we all should learn, particularly plant breeders, more and more to acknowledge the team members. Otherwise, they may not cooperate in future and we may not be able to get the results we are getting. Yes. Then the evolutionary journey of plant breeding was also interesting, the 5 phases. But I would have been happier if you had indicated along with different phases the periods. For example, the first period is over centuries, whereas the latest one is only decades. So that shows how fast we are progressing.
Yes.
That would have given a very good, just one slide. This period, so many centuries, progress is this much. Now, so many years, progress is so many falls. That would have given a good visual information. That would have like. Genomic selection, yeah, it presented very nicely. But what I found, yeah, you gave some examples, but they were not Indian examples in crops. They were all from outside. So that's what I was looking for, an Indian example. And what it is, how it works, you explained. Gene editing, again, you have done a good thing. But I was also surprised. You did not include the example from DRR and IRI in rice. It's a shining example. Last year, the BPT duration was reduced by editing the gene 15 days less duration, yield is same, and I think even quality has remained same. And I have a basmati work again.
Yeah.
By gene editing, that is a remarkable work. I was looking for that, but I mean, I was disappointed that is not shown. And you are a rice breeder throughout your major rice breeder, so I was a little bit surprised. Speed breeding, yes, genetic gain is good and all that. And it has come from Australia because there they have very harsh season. They get only one growing season all along the coast, nothing else. Everything else is desert. So they had to go for greenhouse and all that. And that one breeder was there, Bernard.
Lee Hickey is the breeder.
Yeah, and there are many now. US now many companies are almost all private companies are doing speed
And
breeding.
in UK also.
Almost all UK also same climate problem, so they are taken up. But precision phenotyping is good, excellent, and then using digital means for getting more data in a less time, more precisely. That's actually not a innovation in breeding. It's a method of collection of data we are improving by devices.
Better tool, yeah.
Yeah, so then your concept of digital twins also may work. Still, we have to see.
Yeah.
And this was a new thing for me.
Yeah.
And then the sustainability and regenerative thing also. 6 major challenges how your company is meeting. Here you have come down to discussing how your company, what they are doing. There I was surprised. In crop protection, they are looking for reduction of chemicals. Bayer is a chemical company, basically, like Monsanto or BASF and all that. If they are looking for reducing the chemical, I think they are not meeting their target. Because chemical companies' target is to produce more chemicals, sell more chemicals. Incidentally, they are in seed. But major they are in chemicals.
Dr. Ilyas, I will tell you that most of these companies are now pushing towards biologicals, and they will sell it at higher cost.
Yes.
But they will make the money from biologicals.
Yes, and similar is the objective in direct seeding also. Because along with direct seeding, you have to sell herbicides.
Yes.
So those are linked. I've seen what happens in US.
Yeah.
Along with the seed packet, a bigger packet is of herbicide.
Correct.
So that's going on. And DSR, a lot of work has been done in India, decades in wheat rice consortium.
Yes.
20 years they did all your UP, Bihar and all that area from Punjab. Ultimately, the yield was not achieved, which is achieved under irrigated. Labor was reduced, water consumption was reduced, yes. But yield they could not. And that is the reason I think you cannot get something, hope for something in near future. It may take years.
Yeah.
It will take time.
That is my comment. Precision is good, concept. It's still in US and maybe European country. India, it has not come. We have to see when it comes, how it performs. So overall, it was an excellent presentation.
Yes.
Precise, to the point, within the time limit. So I highly appreciate. I hope all the participants also must have enjoyed this one. So if there are any questions, feel free to ask ATPBR or any of us. We'll be happy to reply. And I finally, before thanking all the organizers, I would again tell the participants, please try to get the publication. It will be useful for your seed production people.
Yes.
Because I have seen right from beginning of hybrid rice till now, almost all the publications on breeding and seed production. So seed production, this type of information is not available at one place.
I hope people will take cognizance of that.
Yeah, so
I only had one clarification. This Precision, because it will base on HDPS, you will increase the density of the plants. So is there an effect on fertilizer application being more, or is there anything done on that?
Thanks, Suren, sir. It is we done extensive testing in the North America and Latin America on all aspects, and overall you can say return on investment of this third corn is better than the other tall varieties, the tall hybrids there, and the absolutely it is having no impact on that front. On additional fertilizer we don't give, because it itself inbuilt you can say physiological efficiency that is making it on that way. And then as you mentioned, Dr. Ilyas mentioned that many of things that you can say on DSR, other things, they are not new. But those are the few avenues where we are investing a lot to see if some differences we can make in the larger front at a larger level. And as I mentioned that it cannot replace TPR in 1 day or 1 year or 2 years.
Yes, yes.
So it will take lots of time and efforts to see. And until unless farmers see the benefit, none of our technology is going to, you can adapt it. So that is where we are also seeing, we are also learning a lot in that journey. Sir. Okay. So let us see how best, and then we require good support from the public sector also in that sense. And we have collaboration, many collaboration with the different organizations also. And then trying our best to, you can say, do whatever best we can do for our customer and environment.
I think many seed companies, multinationals, they are working on DSR.
Yeah.
They are coming out with new genes for herbicide
And
resistance.
new genotypes will emerge probably.
Yeah, new genotypes, and for herbicide resistance also new genes,
exactly.
new herbicide resistant
Exactly. And also probably the IMI, BASF, IMI.
genes. India has become, after IMI, some 3-4 new genes have come.
Exactly.
And everyone is having it one thing. So I would really thank ATPPR for giving me this opportunity, and the speaker, Dr. Shailesh Shukla, Dr. Surendra Kumar Tikku, and
Dr. Ilyas, can I intervene? I have a very short question, if you don't mind.
Okay, please go ahead.
Yeah, actually, this is particularly because the hybrid rice experts are here. Recently, there has been a Chinese report that apomixis has the large potential of revolutionizing seed production, particularly in hybrid rice. What is your comment on that?
See, apomixis is production of next generation without pollination, without sex.
Yes.
So apomictic plants, even if you have apomictic gene, they set only few seeds. They don't set for like a full panicle or something. So that will never be success. And these Chinese have been talking this for last 25 years.
Yes.
Okay. I just wanted clarification on that. Yeah.
No. I think it will not become reality, at least in near future. I don't know in future.
It's not commercial scale. It will take
Commercial scale, it is not possible. As a biological novelty, it is.
Yeah, no, only in fact they have highlighted the commercial impact. That's why I was just wondering, because you are the experts, I can know that.
I would just like to add that, Dr. Gururaj. Suppose, okay, let us say that it will be commercially successful. So what will happen is that there will be very strong implementation of PVP, so that people who are multiplying it will have to pay license to the original breeder.
Okay.
And it cannot be controlled, because once you give it to
that's what, it cannot be controlled.
So where did you see this report? Where did you see it? Where did you see this report?
Okay, thank you very much again, Dr. Shukla, Dr. Ilyas.
Thank you so much.
Very nice. Thank you for coordinating.
Thank you all. Thank you.
We still have 116 people online. That shows how good your lecture was, Dr. Shukla.
So nice of
Thank
them,
you all.
and so nice of you all.
And till we meet again in the next webinar.
And the most important thing is we have kept the time. Total 12:30 and exactly.
Thank you very much. Thank you.
Bye,
Thank you all.
everybody.
Thank you. Bye.
Thank you.
It has been nice experience.
Okay, thank you.